Welcome, future vets! If you've ever stared at a murky jar in the lab and just wondered how on earth that amorphous blob relates to the clean, perfect diagrams in your textbooks, well, you are absolutely in the right place. Today, we're taking those messy, real-life parasite specimens and translating them into crisp, recognizable schematic sketches. Stuff you can confidently reproduce for your practical exams. Okay, let's just dive right into this.
All right, here is our roadmap for today's explainer. We'll cover the metacestode family tree, cystic metacestodes, solid metacestodes, adult cestode heads, adult cestode segments, and finally, a larval versus adult summary. Section 1. The metacestode family tree. Let's start with a big picture to set our biological baseline.
So, before we even examine a single specimen, we really have to nail down the fundamental difference in life stages. Always remember that form dictates the life stage. A metacestode is simply the teenager. It's the larval stage of a tapeworm developing inside an intermediate host. A cestode, on the other hand, is the fully grown, mature adult. So when you're peering down the microscope, the first question you need to ask yourself is, am I looking at a teenager waiting to grow up or a fully armed adult?
It's kind of amazing how neatly we can organize these teenagers. Metacestodes distinctly split into two major visual camps, the solid forms and the cystic forms. When you get hit with a lab question, your absolute first diagnostic step is deciding which of these two branches you're dealing with. Moving to Section 2, cystic metacestodes. Let's dive right into these cystic forms first, which are essentially fluid-filled sacs. When you're trying to translate a murky specimen into your sketchbook, think of Cysticercus overwaters as a simple water balloon containing a single seed. It's translucent, it has a large cavity. Honestly, for your exam, don't overcomplicate it. Just draw one continuous bladder wall containing exactly one single scolex or immature head tucked right inside. Now, contrast that with Coeneris, and this brilliantly illustrates the escalation in complexity. While it's still a fluid-filled bladder, Coeneris is more like a water balloon with multiple seeds. When you sketch this out, draw that same translucent bladder wall, but this time, fill the inner margin with many invaginated scolices tucked inside. Basically, more fluid, more heads. All right, now we reach the hydatid cyst, which is way more complex. This thing acts almost like a microscopic Russian matryoshka doll, literally cysts living within cysts. It's a massive fluid-filled cavity, but its internal structure is highly specialized, and frankly, it's pretty fascinating to draw.
To draw a hydatid cyst accurately, you've got to delineate its specific layers. Start with a thick outer connective tissue layer and the lamellar layer. That's your thick outer cuticle. Just inside that, you'll draw the germinal layer. This is absolutely crucial. It's the hyperactive layer acting as the biological engine that spawns the internal brood capsules. And inside those capsules, you'll sketch the hydatid sand, which are the developing scolices.
Section 3. Solid metacestodes. Let's leave the fluid-filled bladders behind and swing over to the other side of our family tree to check out the solid, elongated metacestodes. Think of these three solid-type metacestodes as your diagnostic checklist. We've got the procercoid, the pleurocercoid, and the tetratheridium. They're all solid, they're all elongated, but they have distinct anatomical features that tell you exactly what stage and species you've got your hands on. Follow these structural steps, and you'll easily identify the solid larval stage under your scope. Step 1. Check the posterior end. If you see a distinct appendage with 3 pairs of hooks, boom, sketch the procerocoid. Step 2. If hooks are totally absent, but there's a holdfast organ at the front, you got the plerocerocoid. Step 3. Look for a worm that's wider at the anterior end with a deeply tucked-in holdfast. If you see that, you're looking at tetratheridium.
Section 4. Adult cestode heads. Moving up to the fully mature adults, let's examine the fascinating weaponry found on their heads, scientifically known as scolices.
Taenia solium is literally armed to the teeth. Notice how the very top portion, the rostellum, features a prominent crown of hooks. But pay really close attention to the shape. These hooks look exactly like heavy medieval boat anchors. Combine those anchors with 4 unarmed round suckers, and your sketch is complete. Let's see how this structure builds with Rhyleida. Both the rostellum and the suckers on this parasite are armed. When you sketch Rhyleida, abstract those distinct hooks into tiny blunt hammer shapes. It gives the head a very distinct, almost heavily textured appearance compared to Taenia.
Next up is Dapilidium caninum. Under the microscope, it has a highly protrusive rostellum. When you translate this to paper, you need to draw multiple distinct rows of very sharp, curved hooks that look identical to a patch of rose thorns. Then, right below that thorny crown, draw your large, unarmed suckers.
So here is the absolutely crucial takeaway. Memorizing these 3 simple visual analogies will guarantee you confidently nail your head identification questions. Is it an anchor? Tania. Is it a blunt hammer? Raletina. Is it a sharp rose thorn? Dipylidium. If you can just remember anchor, hammer, and thorn, you are completely set.
Section 5. Adult cestode segments. Let's move down the body of the adult worm and examine the specific anatomical markers of the segments, or proglottids. Now we're comparing 2 species of Monoesia. At a quick glance, both species' segments are wider than they are long, and both have genital pores that open bilaterally on the edges. But to actually tell them apart on a practical exam, you have to look closely at a very specific marker, the placement of the interproglottal glands.
The only difference you need to capture in your sketch is the distribution of those glands. For Monoesia benedeni, draw the glands clustered tightly together right in the middle of the segment. For Monoesia expansa, draw those dots dispersed all the way across the segment, reaching out to the margins. Center equals benedeni, spread out equals expansa. Honestly, it's just that simple.
Okay, Echinococcus granulosus just breaks all the rules. Instead of a massive tapeworm with endless segments, this adult is tiny. When sketching this one, you'll draw an armed rostellum with hooks, but give it a grand total of only 3 to 5 segments. And make sure you draw the last segment much larger. It covers almost half the total length of the entire parasite.
Section 6. Larval versus adult summary. We've covered a lot of ground today. From teenagers to adults, let's pull it all together. Use this summary to anchor the crucial differences in your mind. On one side, we have our developing teenagers, the metacestodes. They're either fluid-filled cysts acting like single or multi-seeded water balloons, or they're solid, unsegmented larval forms. On the flip side, we have our fully mature cestodes, characterized by their segmented proglottid bodies and fully armed, highly specialized scollices with hooks shaped like anchors, hammers, or thorns. So you've seen the murky, gross specimens, the vivid microscope slides, and the simplified sketches. With these visual translations and specific analogies locked in your head, there isn't a single jar or slide you can't masterfully identify and draw. The real question is, the next time you look down that microscope, what exactly will you see looking back at you? Happy studying, and I'll see you in the next explainer.